And as far as looking at a sagittal cut of the brain, you don't see too much difference between the deprived group and the satisfied group, except you do see more midline activation for the satisfied group and more bilateral activation, especially in the occipital region for the deprived group. This is most likely due to the fact that the people in the deprived group are more aroused in terms of the visual cues, in terms of the visual fields. Their visual fields are more lit up and are more showing more neural activity because they haven't had a cigarette in 10 hours. Seeing a cigarette triggers, supposedly, hypothetically, it will trigger more intensity and a more integrated neuronal network for this group, as opposed to the satisfied group, which you don't see in this part of the brain. You also see a higher amount of activation in the frontal cortex, as opposed to the satisfied group. The satisfied group, when they see that high versus low stimulation, they're not getting the same contrast because they are satisfied. They were able to smoke a cigarette 1 hour prior to the scan. So in terms of their thinking and their anticipation and in terms of their emotional level, they're not going to have as much activation as the smokers in the deprived group would have, which is corroborated so far. And this part of the figures also illustrates what I said before, whereas the deprived group has some bilateral activation in the occipital region versus some midline sagittal activation for the satisfied group. And to look at those activations all together in some glass brains, we put those together in SPM, and really can't tell much right here because it's all jumbled together from all different trials. But you can see that there are certain parts of the brain where the deprived groups would have higher activation than the satisfied groups, whereas once again the satisfied group has more of that mid-sagittal activation. Like I said, I still have a lot more compiling to do, especially comparing the different groups of stimuli and the different categories of stimuli. But I just wanted to briefly mention that there have been a lot of similar studies using PET in the literature. One group in 2002 has already showed that in nicotine-deprived smokers, both the reward and attention circuits, that would be the amygdala, hippocampus, VTA, medial thalamus, were activated by exposure to smoking-related images. In 2002, the group by Brody et al. Showed that brain regions associated with arousal, compulsivity, sensory integration, and episodic memory are activated during exposure to cigarette-related cues and cigarette craving, something that we're trying to show here with using fMRI. And finally, another group showed that results with nicotine influenced brain regions involved, once again, in arousal and reward. And they suggest specific functional systems may be linked to the motivational aspects of tobacco independence, something that I will try to show in the last part of the project with Discuss and how someone can try to wean themselves off independently. As far as similar fMRI studies, which is more relevant to our project, a group led by Smoke-craving et al. In 2005 found that significant correlations between the bold signal, which is what we're measuring in fMRI, and behavioral data, which is really key for our project because we're trying to integrate behavioral data with imaging data. They showed that brain areas related to visual spatial attention and regions involved in motor preparation and imagery were linked together, which is similar to what I showed before with the bilateral activation in the occipital region. And it suggests that the severity of nicotine dependence and intensity of craving are independently associated in separate neuronal networks. This would contradict our initial hypothesis, but this is what they found in their study because we're trying to show that those two networks are in fact linked. Significant positive correlations were found between intensity of craving and metabolism in the orbital frontal cortex and dorsolateral prefrontal cortex and anterior insula bilaterally. If you recall, what I showed earlier was that the deprived group had that significant amount of activation in their frontal cortex. And thirdly, the first demonstration of a greater BTA and nucleus accumbens activation in addicted smokers compared to non-smokers were presented with smoking-related cues using fMRI by a group by David et al. In 2005. So, like I said, I still have a lot more work to do with the imaging analysis. One of the problems with fMRI is that people think that once you're done scanning, the project's over or almost at the end, which in fact is unfortunately not the case, because that's really when the hard part of the project actually begins. Which means that hopefully the next slide I'll present will be another publication by the Liu Lab et al. Showing those linear three-tiered cues, both activation in terms of behavioral data and in neuroimaging data. Thank you very much. Oh, okay, sure.
I'm just sort of curious, maybe another control. What do you think would happen in the deprived group? You gave them a nicotine patch, and then they were imaged. Do they look, like similar to the cigarette smokers, or are they still the same? They just need to see the cigarette.
I think the nicotine patch is something that has effects on a more long-term basis, and this is more of an acute reaction because they're being deprived for just 10 hours prior to scanning. From my understanding, it takes a while for that nicotine patch to have an effect, but I suppose that a secondary follow-up project from the study, which will then be more clinically relevant, you could then compare patients who have been successfully weaned off using the nicotine patch versus patients who, had some other type of deterrent used to wean themselves off the addiction, and then you can compare the two groups. But right now, I don't think that would be relevant, but thanks for the question. Yeah?
Is there anything in particular in cigarette smoke, I don't know if it's nicotine or some other element there, that would have an effect, a direct effect on BOLD, on the vasculature in the brain?
That's a very good question, because the answer to your question is, for nicotine is absolutely yes, because in our other studies, we don't want subjects, smoking at all prior to scanning. So it's obviously a stimulant. It obviously increases blood flow, and that's what the BOLD signal is based on, blood flow in the brain. So, absolutely yes to your question, it does have an effect, but...
So how do you control for that? How do you tease apart genuine neural activity versus this BOLD kind of
I guess, it's a problem. I'm not gonna, try to get around that. But the best answer I can give you is that we have a lot of controls in place in terms of when you subtract one group for another. It's happening to everybody. Well, it's not happening to non-smokers. But because it's happening to all the subjects in that group, you have some consistency there. And that is one of the confounds of this project. So you could then say, well, the people who smoke a cigarette just 1 hour prior to scanning, it's not so much arousal, it's not so much intensity, it's just the fact that nicotine is causing a stimulant, in their blood flow and in their brain. So that is definitely a confound, and there's no easy way around explaining that, except for me telling you that we're being consistent. We're treating everybody the same, we're treating the whole group the same, and that's part of the problem with a lot of fMRI experiments. You can't always get your ideal condition. You can't directly measure what you want, but that is definitely a problem. Thanks. I'm sorry.
Could you get a bunch of people who are all craving for different things and see if there's some craving-specific change?
Not really, because those craving patterns will be different for different things. I guess if it's more drug-related, it'll be a little bit similar, but the craving for food is a different neural network and a different pattern of activity than smoking or alcohol. So, when I used the word craving loosely in the presentation, but really what I meant to say was, craving for a cigarette, craving for nicotine. So craving for nicotine similar drugs, craving a similar stimulant may have a placebo type of effect. But we don't want to be too loosely with the categorization of that emotion.
And I had a question about the 3D rotations. Several times you pointed out that deprived occipital cortex areas were more lateral, and if I remember correctly, that would correlate more to area 18. On the
Yeah,
midline
yeah.
would be area 17.
Right.
And so do you have any idea why you wouldn't have 17 lighting up on the midline as much in the deprived? In other words, it's sort of like splitting.
Right. This brain and this brain are this
And it's
similar.
probably most obvious, let's see.
Right here.
Down at the bottom on those two, that one,
Okay, here we
they're
go.
over there unsatisfied.
Right. So here
So
you.
that's like 18.
And then here you have
or
17.
19, yeah.
I'm just guessing right now. It's something I'm going to look up as soon as I get back. But I would just have to guess right now that that area of activation, be it Rodman's area 18, would be more related to that network of activation in terms of, okay, I see it, and then you're craving it, as opposed to just the actual visual part itself. But the difference between BA18 and BA17 may be linked to, or directly linked to that difference of craving. I haven't thought about that, but that is something I could need to look up.
And then the other thing, the control images, were they, I couldn't really tell, but when I looked at them, I didn't feel like the overall, I don't want to say brightness, but it wasn't exactly balanced. In other words, maybe it was, is the luminance or something of the image equal? Are they equal in all regards? In other words, sort of the span, you have the whites and the.
Oh, there's an equal amount of white, blue, and green. I should have pointed that out. The different color backgrounds are accounted for. This is just one or two examples of
Okay.
maybe 22 different pictures. So
Okay.
I just picked out a couple pictures that I like personally to show.
All right.
But the color, yeah, that's a great question. The color backgrounds in terms of blue, green, and white backgrounds are all equally balanced.
Okay.
Yes, absolutely. No doubt about that.
And then one last one, how did you decide on 10 hours?
That just, in the literature, it just seemed like that was an approximate time of when someone who's an addicted smoker, someone who smokes 6 or 7 cigarettes a day, feels a very high intensity craving for another cigarette. So just based on literature, based on previous non-fMRI studies.
And would you expect if you extended that further, that your significance level would increase between the deprived and the
Yeah, but
satisfied?
we have to have, some, some time period where, we don't want the subject to cheat. We don't want them, to disregard that, the scanning somebody is always, a timing issue, so, I mean, basically it was Dr. Blue's decision, it wasn't my decision, but, whatever, he just said that, that 10 hours was the best
Okay.
time to choose. Oh, one more question, yeah.
For that 10 hours, did you guys do these testings in the mornings or the afternoon?
It varied. It varied between subjects, whenever we could get them into the scanner. Most of them were in the evenings, but.
So it was like all day, a busy work.
Well, we told them, we asked them on the surveys, the paperwork we had, what was your last cigarette? Did you follow up with the 10 hours? And, it wasn't always exact, but, you have, when you're dealing with human subjects, you gotta do the best you can, so. Thank you very much.